Hello everyone, my name is Daniela Herrera, and I'm going to talk about RNA interference, which is a type of technique used in cell biology. RNA interference is also known as double-stranded RNA-mediated interference and is a method used for silencing gene expression in organisms. Silencing is a consequence of degradation of RNA into short RNAs that activate ribonucleases to target homologous mRNA. These short RNAs can be classified into small interferon RNAs, microRNAs, or double-stranded RNAs. Basically, small interferon RNAs trigger mRNA degradation, inhibit translation or transcription of gene encoded in a particular mRNA. So these small interferon RNAs were discovered or first detected in plants that were undergoing either cause suppression or virus-induced gene silencing, and these small interferon RNAs were not detectable in control plants that were not silenced. Then, small interferon RNAs were also detected in Drosophila tissue cultures in which RNA interference was induced by the introduction of long, exogenous, double-stranded RNA. So, RNA interference happens in a two-step mechanism. In the first step, there is the degradation of double-stranded RNA. In mammalian cells, short pieces of double-stranded RNA and a small interferon RNA initiate a specific degradation of a targeted cellular mRNA. And the second step is the formation of the RNA-induced silencing complex, in which the antisense strand of the small interferon RNA duplex becomes part of the RNA-induced silencing complex, which then identifies the corresponding mRNA and cleaves it at a specific site. This technique can be used in functional genomics for the identification and assessment of genes within the genome that are responsible for disease phenotypes. RNA interference is also used for blocking expression of genes and evaluating changes in signaling pathways. Lastly, and I think one of the most important applications of RNA interference is as a treatment model for various pathologies such as viral infections, neurological disorders, and several types of cancers. In the next slide, I will further explain how RNA interference is used as a type of treatment for several types of cancers. The Departments of Gynecologic Oncology and Cancer Biology of the University of Texas MD Anderson Cancer Center have developed a study to analyze the role of RNA interference machinery in the pathways of tumorigenesis and cancer biology in epithelial ovarian cancer and how its expression affects patient outcomes. The name of this study is the Dicer role of Dicer, implications of RNA interference therapy. To answer this question, a cohort of ovarian cancer specimens for Dicer and Drosha expression were used. And in the study, there was a figure that was provided to further understand this mechanism. As you can see on the right side, microRNAs are synthesized in the nucleus. Then it undergoes cleavage to produce short hairpin-shaped intermediates, and then it is further processed by the complex of Drosha and DiGeorge syndrome clinical region gene 8 or DGCR8. Once it is processed, it can be transported transported or translocated to the cytoplasm. This translocation occurs by exportin 5, and then it is cleaved by dicer, which is another type of rRNase 3 enzyme. This cleavage then results in more mature microRNAs. Now, there are other short RNAs like small interference RNAs, which are derived within the cytoplasm and do not require processing by the Drosha DiGiorno syndrome complex region 8. But they do require the cleavage of dicer to become more mature structures. These both when both microRNAs and small interference RNAs are mature, they are activated through the RNA-induced silencing complex, which then produce host mRNA degradation and/or translational repression upon binding on the target. What is interesting about this study is that they were able to confirm that decreased expression of these key RNA enzymes in epithelial ovarian cancers was associated with poor clinical outcome in patients. In fact, the median overall survival of patients with low tumoral dicer expression was 2.33 years versus 9.25 years for those with high expression. These studies were able to be replicated by other scientists, and there were similar findings in lung and cancer patients. However, it was indicated that high dicer expression was a poor prognostic factor in patients with prostate and esophageal carcinoma. And this is due to RNA interference regulatory processes being specific to a type of tumor. And also, it is thought that other components in the RNA interference cascade may also play a critical role in affecting patient outcome. Said this, I have come up with two questions. One is, can this technique be used to analyze antibiotic resistance in different bacteria? And the second one is that the silencing method is a signal. So can this signal be amplified between generations? So this, thank you for watching my video, and here are my references.